Hi friends, good morning. Next video from Dhruva Edutainment and Vidu classes. Friends, this video from Unit 7, that is Organic Evolution of Intermediate Second Year, friends. Friends, in this video we are explaining about genetic drift. Friends, see what is our question, friends? What is meant by genetic drift? Explain genetic drift citing the example of founder effect. Friends, here you have to remember two words. One is genetic drift, another one is founder effect. Already one video is uploaded on this for both Telugu and English medium, friends. This is purely in English, friends. Okay? So what is meant by genetic drift? And explain genetic drift citing founder effect. Founder effect. Now come to definition, friends. See, friends, genetic drift is also called as Sewall-Wright effect or allelic effect. Friends, Sewall-Wright is a scientist who explained genetic drift in the year 1930. So, who is Sewall, friends? He was a scientist. He explained about genetic drift. Because of that, we are calling genetic drift as what, friends? Sewall-Wright effect. Next, allelic effect. So, alleles. What are alleles, friends? A gene has two forms. They are called as what, friends? Alleles. So, here the frequency change in the population. Because of that, it is called as allelic effect. Now, see, friends. First, you take the definition. Change in the frequency of a gene or allele. You have to write any one of these two, friends. You know, friends, majority of the genes have two alleles. Change in the frequency of a gene or allele in a population by chance, not by natural selection, is called genetic drift. Friends, here you remember three words. Change in the frequency of allele by chance, not by natural selection. Friends, change in the frequency of a gene or allele in a population by chance, not by natural selection, is called genetic drift. Friends, here population means here you have to concentrate on small population. Genetic drift occurs in small population. It won't occur in huge population, friends. Okay, friends, it occurs in small populations only, not in huge populations. Okay, friends, that is about the definition, friends. You know, friends, generally a gene has two alleles. Forget about the multiple alleles, friends. But majority of the genes have two alleles only. So, a gene has what, friends? Two alleles. What are alleles? Two forms of a gene is called as allele, friend. See, friends, if the frequency of one allele is 1%, the probability of losing that allele is more from a small population. See, an allele has less frequency, friends, that is nearly 1%. So, the probability of losing that allele is more from a small population. If an allele has more percentage, if the frequency of allele has more in a population, so it won't eliminate, it won't lose from that population. If the frequency of one allele is 1%, the probability of losing that allele is more from a small population. That is genetic drift, friends. Genetic drift may result is either fixation of allele, friends, fixation of allele in the population or loss of allele from the population. So, what is the result of the genetic drift, friends? Genetic drift may result either fixation of that allele in the population or elimination or loss of allele from that population. See, friends, what is fixation, what is loss? A gene or allele permanently established at 100% frequency in the population. Please don't write this in your IP, friends. What is fixation of allele, friends? If a gene or allele permanently established at 100% frequency in the population is called as fixation. What is loss, friends? A gene or allele permanently lost in the population is called as loss, friends. So the alleles which have less frequency may have chance of eliminate from the population. The alleles which have more frequency may continue their fixation, may continue in that population, friends. Now, genetic drift tends to reduce the amount of genetic variations. See, friends, genetic variation in the genotype. Genetic drift tends to reduce the amount of genetic variations within the population, mainly by removing the alleles with low frequency. So when low frequency alleles are eliminating from the population, that character is also eliminated from the population. So because of that, what happens, friends? Genetic variations are reduced in the population. Genetic variations are reduced in the population due to what, friends? Due to removing of the alleles which have low frequency, Now come to founder effect. Friends, genetic drift explained with two examples, friends. One is founder effect and the other one is bottleneck effect. Here you have to write about the founder effect only, founder effect. See, founders, first you know what is the definition of founders. Founders means a group of small animals which separate from main population and form a new colony in isolated area is called as founders, friends. See, friends, when a small group of individuals from a population, friends, when a small group of individuals from a population starts a new colony in an isolated area. So here, this small group of people are separated from the main group and they are forming a new colony in an isolated area. These small group of people are called as what, friends? Founders. Those individuals are called as founders of the new colony or new population. So here these people who are separating from the main population, they are forming a new colony in an isolated area. These people are called as what, friends? Founders. These people are called as what, friends? Founders. Now, new populations is genotypically and phenotypically. Now there is a new colony, friends, in an isolated area. They have their progeny. So the progeny resembles what, friends? The progeny resembles the founders, not by this main colony people. Okay, friends. New population is genotypically and phenotypically similar to these of the founders, but different from parents. Parents means ancestral population, friends, from the main population. So, new population of the new colony genotypically and phenotypically similar to those of the founders, but differ from what, friends? Different from parents or ancestral populations. Examples, friends. O blood group is present in nearly 100% of red Indians. See, red Indians have O blood group. Red Indians have O blood group, friends. This is MSET question, friends. What type of blood group is present in red Indians? That is O blood group. It means the forefathers of red Indians were predominantly O positive. And they isolated these red Indians. Majority of the forefathers of the red Indians have O positive blood group. The people who are having O positive blood group are separated from the main population and they establish a new colony in an isolated area. The people who entered into that new colony have O positive blood group. So now what happened, friends? The progeny of the offspring of the new colony all have what, friends? O positive blood group only. See, the forefathers of Indians were predominantly O positive and they isolated themselves reproductively and started a new population. See, friends, here one example. See, this is a main population, friends. This is a founder. This founder separated from the main colony and it established a new colony. See, in this new colony, all organisms have the same character of the founder. This is called as what, friends? Founder effect. See here in the main colony there are other genotypes and phenotypes, friends. Morphologically and genotypically they are dissimilar. But here in the new colony, in the founders colony, genotypically, phenotypically they are all same. This is called as what, friends? Founders effect. Founders effect. Friends, this is about what, friends? Genetic drift. Friends, genetic drift. This is purely for English medium people, friends. Okay. Thank you. Good day.